This podcast is for general informational, educational purposes only and is not to be considered medical advice for any particular patient. Clinicians must rely on their own informed clinical judgments when making recommendations for their patients. Well, welcome everybody to another episode of PHM from Pittsburgh. I am still your host, Dr. Tony Tarchichi. If you're new here, welcome. I'm a med-peds trained pediatric hospitalist here at UPMC, Children's Hospital of Pittsburgh. We have had this little podcast now since October of 2016, giving you different topics in pediatric hospital medicine, trying to go through all of our content specifications. Like everything else in this world, COVID has thrown a bit of a monkey wrench into what we normally do. We have had the opportunity to cover quite a few topics in COVID, and today is no exception. With the pandemic still raging, cases rising everywhere, and if you're listening and you're a physician, I wish you the best of luck. We're all in this together. We're all working very hard. Stay safe. Take care of yourself. We are getting closer to what is going to be a vaccine, ideally, and healthcare professionals, according to CDC, are the first to get it. So we have naturally brought up a lot of questions of where are the vaccines from? What do they actually say? Can we get into some specifics? Especially because our patients are going to look to us for this information. And as of yet, I don't really have that, besides what I've read on CNN or NBC or Fox News or wherever you get your news. So I thought, I need some help. So whenever I need help with an infectious disease topic, I've turned to two people. And I'm fortunate enough that both of them are here to help us out with this conversation about the COVID vaccine. We will start with Dr. Megan Culler Freeman, who's now a friend of the podcast, been doing this a while with us. Dr. Freeman is currently a senior pediatric infectious disease fellow at the UPMC, Children's Hospital of Pittsburgh. She completed her MD and Ph.D. degrees at Vanderbilt University, where she earned her Ph.D. studying coronaviruses in the laboratory of Mark Dennison. She has multiple publications in the field and is the author of the coronavirus chapter in the most recent edition of Nelson's textbook of pediatrics. Her current research interests include pathogenesis and cell biology of RNA viruses and advanced tissue modeling, with a current focus on enterovirus D68, the hypothesized cause of acute flaccid myelitis in children. And Dr. Freeman, thank you very much for joining us again.
Thanks again for having me.
And of course, friend of the podcast, honorary co-host, Dr. John Williams. Dr. Williams, a professor in the Department of Pediatrics at the University of Pittsburgh School of Medicine. He's the chief of the Division of Pediatric Infectious Disease here at UPMC Children's Hospital of Pittsburgh, and the director of the Institute for Infection, Inflammation, and Immunity in Children, or I4Kids. John, thanks for joining.
Tony, it's always a pleasure to be with you.
All right, so let's get our disclosure statements done and out of the way. I will go first and kick it out to you, Megan, and then last but not least will be John. So I have nothing to disclose.
I also have nothing to disclose.
I serve on the Scientific Advisory Board of Quidel and of ID Connect, and I serve on an independent data monitoring committee for GlaxoSmithKline, neither of which is related to COVID or to the topics under discussion today.
All right. Thank you, John. So I thought we would start broadly and then get into more specifics for these COVID vaccines. So first of all, like most people, this is the first time I have ever paid this much attention to vaccine development in my life. And I'm a pediatrician who loves vaccines, and I support vaccines. I think the whole world's in this boat. When researching for the podcast episodes normally, and you both know this, I read numerous articles. You send me articles. I send you articles, and when we talk, and then I'll go through them all. Sometimes I start with textbooks to make sure I have a reasonable understanding of whatever it is we're talking about. Here, it was totally different. I found two articles that were of any substance, and the rest is press releases from two pharmaceutical companies. So how abnormal or normal is this for vaccine development?
Well, I'll take a start on that one. It's in the sense of the release of the data, it's actually not unusual. So although, Dr. Freeman and I also right now really only have access to, the scientific data from animal studies or whatever's been published on very early studies. And then we have the press releases like you. But we all have to remember, this is not quite the same as, a company who says, hey, we are selling a new form of Tylenol, and we think you should buy it. So for any kind of clinical trial, a drug, a vaccine, anything else, there's what's called a Data Safety Monitoring Board, a DSMB. And this is a panel of typically scientists and doctors. They might be, in this case for a vaccine, ID doctors, virologists, epidemiologists, and they're the only ones up until the announcement is made that actually see the unblinded data. So the executives of the company, the people doing the trial, they don't know who's getting vaccine and who's getting placebo. And those reports go typically every month or more frequently sometimes to the DSMB, who, in a private meeting, review those data, and only at a point where they can see there's something happening, then do they release it to the company to say, we think you can unblind it. And so, although all of us have, even as scientists and doctors, we just have the press releases, it's actually been through a panel of experienced doctors and scientists. So that gives me a lot more confidence in the results than if it was just the company's opinion, hey, we have this great new thing.
Now, John, I want to follow up on that because I think a lot of people are skeptical of this process, how quickly it went and how quickly these vaccines turned out. So the DSMB, the Data Safety Monitoring Board, these are very experienced clinicians. These aren't people they just grab who are yes men or yes women in terms of really pushing this through. These are people with a lot of experience and will push back to the company and say this isn't working or this is a bad idea or this is blah, blah, blah. Is that right?
Yeah, so a couple of things there that, in terms of, designing and running the trial, companies work with the FDA early on in the design of trials. I mean, the FDA wants things to be done right and to be done safely. So it's not in the FDA's interest. I mean, they're funded by taxpayer money. The FDA is there for us. And so it's to our benefit and to the FDA's benefit to work with the company to make sure they do the trial right. And so even before a DSMB gets involved, the FDA is telling the company, essentially it's more formal than this, but the company says, hey, we think we need to do this many people and do it like this. What do you think? And the FDA looks at their plans and says, no, you need twice as many people. And the company's like, okay. So the design actually gets vetted. Usually what takes so long is the company takes, 2 months to generate this 150-page massive document. They send it to the FDA. Like all good government agencies, it takes 3 months to review that giant document and send it back for revisions. Everybody worked a lot quicker this time. So that was the first thing that happened a lot faster. Once the trial starts, the DSMB absolutely has the power, and I've done this in trials that I was on the reviewing committee for. The committee can say, wait a minute, we have a subject who had an adverse event and we need to halt enrollment and we need to know more about that person to know, is their adverse event, maybe they got hospitalized, is that related to the product they got? And the company still doesn't know what group the person's in. And they go, okay, we'll halt enrollment. We'll give you whatever data you want. And the board reviews it. And then they say, oh, it's not related. Okay, carry on. And the trial resumes. So people, your listeners might remember that happened, right? With AstraZeneca back in the summer. They halted the trial and the investigators determined, the board, the safety board determined, not related, you can resume. So it happened quickly, but in this case, quick actually doesn't mean bad. And I feel confident of that in this case because I know how the process works and they just did the really slow bits faster. And then, tragically, there's so much COVID right now that they didn't have to wait. We don't have a lot of tetanus in this country, right? About 500 cases a year. So in unvaccinated people. So if I want to test a new tetanus vaccine, man, I'm going to have to wait a long time to get enough cases. But because people are being so foolish about not wearing masks, we could rack up 100 cases of COVID really quickly. So sometimes faster means not as good. I truly don't think that's the case here.
Well, that's good to hear. Now, Megan, I'd love to kick out a question to you, and this is your area of expertise. So could you please review for our listeners and for me the virology of coronaviruses before we start just to give us a refresher framework in terms of the virus itself, so when we're talking about different aspects of it.
Sure. I will focus mostly, since we're having a vaccine conversation, about what your immune system is going to see when the virus infects you. So these are RNA viruses. They're pretty large for their family of viruses. And everyone at this point, I've never had an audience that is more familiar with the coronavirus than I do now. So everyone at this point has seen a nice diagram of a spherical virus that has some protein sticking out of the membrane surface. That protein is called spike, very aptly because of its shape. And the two vaccines that we're probably going to focus on today, which are the Moderna and the Pfizer vaccines, that are the ones that have completed their phase 3 clinical trials in the United States, both focus on that spike protein. So the idea being that if your body had antibodies that would be able to bind to that spike protein, that perhaps it would be able to neutralize the virus before it had the opportunity to interact with a cellular receptor on your cells in your nose or your lungs or wherever else in your body. And that way the virus would not be able to get into those cells, replicate, and cause disease.
Now, if I understand it correctly, Megan, the spike protein binds to the ACE2 receptors. Is that only in the lung tissue, or is that if you swallow it, will that go in the esophagus, for instance?
Turns out that ACE2 is in a variety of tissues in the body. It's really highly expressed, like you were saying, in lung tissues. It's also in the endothelium of the blood vessels. There's pretty high expression in the kidneys, and several other organ systems. So any place that ACE2 is available, I think SARS coronavirus 2 has probably found a way to use it.
Okay. All right. Now, from the paper I read, and the paper is SARS-CoV-2 vaccines in development. It's from Nature, September 23rd, 2020. And if I understand this correctly, natural infection from coronaviruses induces both a mucosal antibody response, which is for those listening, that's secretory IgA, and a systemic antibody response, and that's, your IgG. And the upper respiratory tract, we know, is mainly protected by the IgA. In the lower respiratory tract, the IgA immunoglobulin, and the lower respiratory tract is mainly protected by IgG. All the vaccine candidates in production are intramuscular, intradermal vaccines, and they typically only induce an IgG response. This paper makes a large point of this and says mucosal IgA immunity might not be required to protect from severe or even symptomatic disease for that person. So how much does it matter or not matter if we do not have an IgA response to any of the vaccines in production for our patients or public health or anything? We'll start with Megan. We'll start with you, then go to John.
Absolutely. So that response of immunity that you describe is pretty common for a lot of respiratory viruses. I think RSV is the one that we think about a lot. So we know that young children have a lot of trouble, lower airway disease with RSV, and then are able to make antibodies in the short term, IgA, and in the longer term, IgG. But as they get older, we're able to be reinfected with RSV because our mucosal immunity has waned. But when you get older, you don't have severe disease. You have a minor cold. So that's a pretty similar thing that I would perhaps expect to happen with coronaviruses as well. So it's great that we would be able to prevent either symptomatic disease or severe disease. And I think as a goal for vaccine development, that seems like a very worthwhile goal because that's really what's killing us with this. I didn't mean to make that kind of lightly, but with this virus, right? It's literally killing people. It's quite severe in a very large segment of the population comparatively. So if we can prevent severe disease, that is quite the achievement. The thing that does worry me a bit is because this virus is so competent at replicating in the upper airway. And we know this because even asymptomatic people can spread it from one person to another when they're having a conversation without a mask. It does concern me that if someone was vaccinated and protected from disease, but not protected from infection, perhaps they could have replicating virus in their nose or their upper airway, and they could spread virus to another person who perhaps was not unvaccinated, and they could still have a pretty severe outcome. So that is, I think, what we're all waiting to see, kind of what the data is going to be in that respect. But that is a potential challenge that we might be looking forward to.
Yeah, I think, Dr. Freeman really hit the key points there, Tony. So, to RSV, which is, I think, the virus that we know the most about in terms of this serum versus mucosal immunity, I hope you have listeners from Rochester who know of or knew the late, fabulous Karen Hall. So, Carolyn Breesy Hall. Karen was a world leader in RSV research, a PEDS ID doc at Rochester for her whole career, basically. And Karen did this wonderful, wonderful study many years ago where she took pediatric nurses. Okay, so these are nurses who have a very high serum IgG to RSV because they're getting boosted constantly by their patients, right? And she took one stock of the virus, not mutated or drifted at all, the same exact stock, and squirted it up their nose at time 0, 2 months, 4 months, 8 months, 12 months, 24 months, something like that, okay? And actually, the first interval was 1 month, 0, 1 month, 2 months. Even 1 month after they had been just infected. Okay, so all these nurses have high IgG in the blood. Half of them became reinfected and were able to shed virus, like Megan was talking about, so they could potentially transmit. And half of those people were symptomatic. Now, it was a mild cold, and that continued throughout the study. So you could challenge these, highly immune nurses over and over and easily reinfect them with RSV, and they could shed some virus. So it's not at all crazy to think, well, will the vaccine prevent transmission? We're not quite sure. It's likely to lower transmission because if you have some immunity, adults get reinfected with RSV and metapneumovirus and flu, but we shed a lot less virus for a lot shorter period of time. So we're not nearly as contagious as kids. So it's likely to decrease transmission, even if it doesn't totally block it. But I think the point that Megan made about disease severity, that's really the key. Nobody's up in arms about rhinoviruses, even though, it's the most common respiratory infection humans get. And even right now, during the pandemic with mitigation, we're still seeing tons and tons of rhinovirus infections. It's not really killing anybody. Okay, it's just not that severe. So that makes it kind of less important in a way. And a lot of vaccines, some really prevent disease. MMR, varicella, you're not going to get measles if you're exposed. Tetanus, you are not getting tetanus. But flu vaccine often doesn't totally prevent flu, but it lessens severity. It turns out some immunity is a lot better than none. And the Moderna trial has seen this, that there was not just less COVID, like all comers, like COVID, yes, no, but there was less severe COVID. And just as you said, Megan, fewer people dying, huge win. Huge win.
But so if I understand this right, if you get the vaccine, you're protected against severe disease. You may get a mild case. If you get infected, it's very possible, but you can still transmit. So is this what I've been following Dr. Fauci, like everyone else in the country has been following Dr. Fauci? Is this what he's talking about when he says it's going to be a while before we get back to normal? Because we will have to vaccinate a lot of people and everyone will have to take the vaccine to really cut down spread and maybe get rid of this virus altogether. Those who are, let's say for us as healthcare professionals, if we're vaccinated first, we could still get the virus. It won't cause any real issues with us, and we could still spread it to our loved ones at home or people in the supermarket if we're not masked. Is that right? Is that what I'm hearing?
That's potentially what could happen. I think the other bit of that is it's just going to take time to vaccinate a whole world worth of people. So even if you're in one of those first waves of people that are able to be vaccinated, there's still going to be a majority of the population that needs to keep masking and distancing and everything else. And, as a pediatrician, I like to set a good example. So even after I'm vaccinated, I'll definitely still be wearing a mask out in public.
Yeah.
Well, yeah, and that's a really key thing is that because there is, we don't know what the number is yet of transmission. If I'm vaccinated, what is the chance I'm going to get mildly infected and pass it on? Is it 50%? Is it 1%? But it's something. It's definitely there. We know people can get reinfected, right? So we know that the immunity isn't 100% protective. And this is really important for people to know is that even though they get vaccinated, they will still have to mask. Just as Dr. Freeman said, the three of us, whenever we get vaccinated as healthcare providers, we'll still be masking.
Now, here's one of the technical areas I'm going to need help in. Pfizer and Moderna's vaccines are messenger RNA-based vaccines. They're the ones that have been in the news all the time. What is that? I know what messenger RNA is in terms of function in a cell, but what does it mean to have a messenger RNA vaccine, and how different is this in terms of what it's targeting than established vaccines like Tdap, like Rota, like even the flu that we already know about?
This is quite a clever vaccine platform, actually. So it is an mRNA, so the message that your cells know how to use and translate into a protein that then can be expressed by your own cells. And it is in kind of a lipid substance that is part of the injection as well. So what's going to happen is that's going to be taken up locally by cells in your deltoid where you get your injection. And some of those muscle cells might be transformed or transfected in a way that they are able to then utilize that mRNA as the message and make that portion of the spike protein that then is going to get presented to your immune system, and you're going to be able to form antibodies against it. There's also probably going to be some kind of minor amount of cell death at the site that will then get gobbled up by dendritic cells and other immune cells that are there, and that's going to contribute to that antigen presentation as well. This is a new area, but I have been doing some reading that vaccines that are made in this way, as opposed to injecting a preformed protein, have a greater propensity to make a balanced T helper cell response, which is what you want in a vaccine. You want a balance between Th1 and Th2.
Why do you want that, Megan?
Well, there have been historically some issues with vaccines in the past that have a Th2 shunted response. And these are the issues where we see, in particular, the RSV vaccine from, I think, the 60s is the one that comes to mind, that those infants, unfortunately, when they got that vaccine, their immune response was shunted towards the Th2 instead of being balanced. And when they were exposed to RSV, they actually ended up with worse disease instead of improved outcomes.
So,
the other thing,
John, did you want to say anything?
Yeah, just because the other thing that's really clever about these vaccines, and I think it's going to change the way we make flu vaccine. So, flu vaccine, it's a killed virus. People grow the virus in eggs or in cells, and then they chemically inactivate it. And one of the flu vaccines is synthetic protein, produced in cells, and it's a flu protein called hemagglutinin that is functionally the cousin of spike. Okay, it's very similar to spike. And so flu vaccines work. They don't induce this bad response that Dr. Freeman just referred to. But, flu vaccine is a lot better than no flu vaccine, but it's not great. So the other really clever thing of these two vaccines from Moderna and Pfizer, these spike or hemagglutinin, this family of proteins, has two different shapes, a pre-fusion shape and a post-fusion shape. Both work, but the pre-fusion for other viruses is way better. Now in nature, with like a killed vaccine, you have mostly post-fusion, which works, but it turns out the pre-fusion is a lot more potent. So I think a lot of us think that in a couple of years, we'll have new flu vaccines that are a lot like these COVID vaccines, and we'll go from 60% effective for flu vaccine to 90% effective.
Oh, now that would be something.
It would be huge.
To explain the fusion protein a little bit more, so the spike protein's job is to pull the membrane of the virus together with the membrane of the cell so that they can fuse. So this protein has the situation where it essentially will kind of jackknife itself into the cell and then completely rearrange itself so it looks different as it's kind of acting as a mechanism, a physical mechanism to pull the two virus and the cell together. So the shape of the protein looks very different post-fusion than it does pre-fusion.
Oh, okay. Now, so then, from what I have read, we need 2 doses of these vaccines about 21 days apart. Why do we need 2 doses?
I'm not sure that they revealed their data after 1 dose, but oftentimes we need kind of an initial exposure to an antigen and then a booster to really make sure that we have an adequate immune response. I think time will tell if some of these vaccines are able to get away maybe with 1 dose instead of 2, which would certainly have kind of distribution benefits because it's going to go twice as far and maybe happen twice as quickly that we could vaccinate everyone. But I don't know if they evaluated 1 dose as a primary endpoint.
Yeah, I mean, I that data is not yet available. I would guess that they did, and that after a single dose, there just wasn't that much of a response. This is one thing that, for a non-live vaccine, whatever format it is, if it's not a living organism, a live vaccine like the flu mist, like MMR, like varicella, right? You're having a mild infection. So the virus is hanging around for a number of days, and it's inducing innate immune responses, and it's driving the whole process. With any sort of, not living vaccine, whether it's a killed or a synthetic or a toxoid like tetanus, diphtheria, typically the first dose primes the immune system. So it's all ready to respond, and it makes a little response, but it's just not that much of a response. But when you give a second dose, boom, then you get a really, potent response. And that first priming dose doesn't have to be all that much. And so, this is why in young infants in particular, this is true. Like, this is why they get repetitively vaccinated. But if you had an 18-year-old who had never had, any vaccine at all, I mean, they're not going to be all that protected after their first DTaP. They're going to need really a couple to really get good protection. So they may be able to get around this with different adjuvants, but I think the current vaccine probably going to continue to need the 2 doses.
And just so our listeners know, all the data we have to go on is from press releases. The raw data from Pfizer and Moderna has not been released, like John and Megan were alluding to. But we have heard that Pfizer will release its raw data for its patients, at least as far as I know, in a few weeks, from according to the CDC. So we'll keep an eye out for that.
For most, vaccines, drugs, anything, Tony, once it goes through the FDA process, then most of that data becomes public. I mean, not to the level of individual participants, obviously, still HIPAA protected. But yeah, the reason it's sort of legit. It's like filing for a patent. Like, you keep it secret when you're filing for the patent, but once the patent's approved, then it becomes a matter of public record. It's a little bit like that. So yes, we should have access to all of this data. And I know there's been a lot of buzz out there in at least the lay press and the lay media about, oh, is it safe? Is it safe? People should not forget, and we as physicians, and a lot of your listeners are healthcare providers, obviously, we should remind our patients and our friends and families, look, every drug, vaccine, product approved by the FDA has phase 4, which is post-licensure surveillance. Okay, so companies have to continue monitoring. Okay, we did it in 30,000 people. It looked safe, so it was approved. But we got to watch the first, million or whatever to make sure it's safe. And a number of things have been pulled in post-licensure marketing, including the first rotavirus vaccine, RotaShield, 2000. It was licensed and it was pulled less than a year later. Merck had a big COX-2 inhibitor that was pulled in post-licensure surveillance because it turned out to be related to increased risk of stroke.
Now, Megan, and this SARS-CoV-1, what you did your Ph.D. in, I had read that they had begun phase 1 trials on a vaccine for, but because the virus really just disappeared because it was so different, that stopped. Is that right? And was it a similar vaccine, this messenger RNA type thing?
So SARS-1 happened in 2002, 2003. So it was not a similar platform vaccine. The mRNA vaccine technology is a lot newer than that. But you're exactly right. Kind of the existence of SARS-1 and the wild completely evaporated. So then it was not even possible to test the vaccine for effectiveness. Some of the background work that's contributed to both the mRNA platform and coronavirus vaccine was actually in MERS, Middle Eastern Respiratory Syndrome coronavirus, which is still circulating, although at a much lower level than SARS coronavirus 2, it's been completely eclipsed. But there had been a lot of preliminary data with similar vaccine platforms to the mRNA that we ended up using for SARS coronavirus 2 in that virus.
All right. So now in terms of side effects, which is what everybody wants to know, whenever you read any blog, news site, comment section, or just talk to anyone, what are the side effects? I have read that most side effects to vaccines in general occur within the first 45 days of getting a vaccine. Now, first, before I continue my question, is that right?
It's going to happen pretty close to the immune challenge. So I think that makes complete sense.
All right. And so that's why Pfizer and Moderna took the phase 3 trials out to 60 days because they wanted to make sure to capture as many of the potential side effects as possible. Is that true? You agree with that? You weren't in the meetings clearly, but
I wasn't there, but it seems like a reasonable strategy.
all right. And on the Pfizer press release for the phase 3 trials, they say data, and I'm quoting, data demonstrate vaccine was well tolerated across all populations with over 43,000 participants enrolled. No serious safety concerns observed. The only grade 3 adverse event greater than 2% in frequency was fatigue at 3.8% and headache at 2%. So first, can you explain this grading system for adverse events?
Sure. So the FDA divides events into mild or grade 1, moderate, grade 2, severe, grade 3, or potentially life threatening, which is grade 4. And pretty much for everything that you could think of that might be related to a vaccine. So things like fever or diarrhea, headache, fatigue, myalgia, pain at the injection site. They have a scoring system that would correspond to each of those grades. So if it's so mild that perhaps you notice it, but it doesn't interfere with your daily activity at all, then that would be a grade 1. If we're thinking about headache, for example, then if it's a grade 2, maybe you needed to take an NSAID or other types of pain reliever. And it interfered a little bit with your activity, but it wasn't that big of a deal. You could still go to work and that kind of thing. That would be a moderate or a grade 2. Anything that's in the grade 3 or severe category, usually they will call as something that has significant interference with your daily activities. So that sounds like those fatigue and headache that they described in the minority of patients perhaps were in that category. And then the grade 4 involve at least a visit to the ER or a hospitalization event.
Yeah, and I think it's really important to emphasize that, you know, these are skewed towards, especially for vaccines, okay, right? Because the whole point of any vaccine is you're giving it to somebody who presumably is healthy, or they may have other underlying health conditions, but if you're giving a vaccine for tetanus, you're not giving it to somebody who has tetanus, right? You're giving it to somebody who is not infected. So safety always takes top priority. In fact, in the phase 1 trial, it's really about safety. It's not about does it work, it's about is it safe. And so grade 3 is called severe, but for example, I got my shingles vaccine a couple of months ago. I believe Megan may have heard me complain about this. And it's the Shingrix, which is the newer version, which is way, way better than the older version. It's much more effective. And it's got a novel adjuvant, you know, a fairly recent modern adjuvant that's more potent. The next day, I had a significant fever, terrible headache, chills. I mean, I felt terrible. Like, it's a good trade-off because I don't want to have shingles. I've seen shingles. It's much worse. But, you know, most days I come into work. That day, I actually, you know, stayed home and worked at home. I still worked. I didn't go to the doctor. But the fact that I chose to stay home, grade 3, severe. But if you said, did you have a severe side effect? I would say, no, that's not, you know, my arm hurt and I felt crummy, but I wouldn't call it severe. So I do think people need to keep in mind these adverse effects are very formally scored and graded, but a lot of things that are severe, as Megan said, fatigue and headache, those people probably had like what I had with my shingles shot.
In terms of how these companies would make these press releases, and you may not be able to answer this, and I don't know if I'm going to formulate a good question or not. If there was a grade 4 event, I'm assuming they would have to put that forth in their press release, or could they hold that back?
No, that's a great question, Tony, is because I think people often worry, can companies hold any of these things back? And the short answer is no. So companies, I mean, would get in dire trouble, you know, dire trouble, like $1 billion penalties if they held back data from the FDA. And so they're filing to the FDA. I mean, the final documents they turn in are like, God only knows how many pages. You know, it's huge. Um, and it includes all of this stuff. And it includes both solicited. So I ask, did your arm hurt? Were you tired? Did you have a headache? And unsolicited, hey, did you have anything? And if you, Tony, felt dizzy, and I didn't ask you that, you would say, yeah, I felt dizzy. And I record all of that because, well, what if 20 people complain of things I don't ask? And everything is recorded. So if a vaccine recipient gets in a car crash on the way home and they go to the ER, that's a grade 4. That had nothing to do with the vaccine, but that's recorded. Every health event is recorded and scored. And because, again, the focus is to not miss a safety signal. And like drug companies aren't, just in case some of your listeners have heard this, number one, drug companies aren't stupid. So they're not thinking, hey, let's sneak this one through the FDA and, we'll get our vaccine out and then it'll get yanked 6 months later. Like that won't make us look bad. And also, okay, vaccines are 2% of Big Pharma's portfolio. So nobody's getting rich off vaccines, okay? They make Viagra too. Viagra is a $12 billion a year drug. All of Pfizer's vaccines together are probably less than $1 billion. So there's not the financial incentive for vaccine companies to be sneaky and hide these data.
But now, what about in these press releases you're talking about to the FDA? In these press releases they release to us, if there was a grade 4 event, would they have had to release it, or could they have waited to just tell the FDA, or do we not know?
Well, I don't think they would have to say it in a press release, right? I mean, in a press release, sort of the definition of a free press, which I believe in, you could sort of say anything you want. But in this case, I think, Tony, because it's going immediately to regulation, to the FDA, I think there's no value. Now, do companies make misleading or false claims in press releases? Yeah, all the time. Like look at all the COVID tests. And you see all these ads for COVID tests, home COVID tests. Newsflash, those are all fake. Either the test is never coming or it's not any good. And those aren't regulated, right? So the company can say, our COVID test works at home and it costs $5 and it's awesome. And if 1 million people pay $5, they just made 5 million. But yeah, in this case, are they obligated to say it in the press release? I don't think so. But I truly think there's no reason for them to hold it back because FDA is going to know, and they're only hurting themselves.
Now, the reason I'm asking to listeners who normally listen to me, I don't normally ask these types of paranoid questions, but I think these are the type of questions patients are going to ask you because these are the type of questions my family's asking me and patients are asking me. So I'm trying to prepare us for when patients start asking these things. And I'm assuming the answer to my next question is we don't know was there any other adverse events recorded? So like, Megan, you were talking about grade 1. That could be like if you have a rash at the site, which you don't even notice, but it's there. That could be a grade 1 reaction. I'm assuming that's recorded, but we don't know that information yet, how many of those types of reactions there was.
Yeah, that's exactly right. For rashes, they actually have a whole scoring system based on the diameter of the rash. So,
is that right?
Yeah.
Oh, wow. All right, so now let's focus a wee bit more on Pfizer and Moderna's vaccines since they're both undergoing review for the emergency authorization currently by the FDA. So Pfizer's vaccine, they said, is 95% effective against COVID beginning 28 days after the first dose. And Pfizer enrolled 42,722 participants in the study who received 2 doses. 170 had COVID disease. Confirmed. 162 of those were in the placebo group and 8 were in the vaccinated group. So what does this mean? What do all these numbers mean to me as a pediatrician talking to patients? Does that mean if I get the vaccine, then 28 days later, if I'm exposed to SARS-CoV-2, 95% of the time, I won't get it at all?
I think that's a pretty good interpretation.
Yeah, I mean that's sort of ballpark what it means. In a trial, they study vaccine efficacy, which is it's a controlled setting. Everybody, they know if they got vaccine or placebo, and they're tracking them all for disease. And yeah, essentially, I mean, the example I use, I take even simpler math. Okay, you have 100 people in each group, and all 100 people in the placebo group get infected and only 5 people in the vaccine group get infected. That's 95% efficacy, right? Because you just reduce cases by 95%. In the real world, post-licensure, and here at the Children's Hospital of Pittsburgh and at the University of Pittsburgh, we're a site to do studies in adults and kids every year to look at the effectiveness of flu vaccine. Effectiveness means basically like out in the real world. And the way we look at that is that same sort of way. So if we say, oh, flu vaccine this year against the circulating strains was 65% effective in kids, that means it reduced 65% of the disease, which is huge. When you think about, 20 million kids get the flu every year in America. 20% of the kids in America, so about 12 million kids get the flu every year. So if we could reduce that by 60%, that's huge.
So that's what you mean by reduce. You mean the number of kids who actually get the flu and are tested flu positive decreases by that amount of number because of that vaccine.
Yeah. Most these trials, as we'll touch on this in Moderna, but these trials and most vaccine surveillance really focuses on disease, yes or no, because it's easy to focus on that. Did you get sick or did you not get sick? It's a lot harder to focus on severity. Like Tony, you and I, we get the man flu. So we stay at home whining and griping and moaning. We're like, oh God, I had such severe flu. Megan, of course, goes to work, and she's like, oh, it wasn't that bad. And actually, if you measured it, maybe she had worse flu, but you and I made it seem more severe. So severity is a lot harder, measure to get a hold of. So most of these studies look at just, disease, yes, no.
I think the other thing that's interesting to think about is these are people most likely that had symptoms that led them to be COVID tested, and then they discovered that they had a confirmed case of COVID. They were not going to everyone every day, all 43,000 participants, and swabbing their nose to see if they had acquired infection. So it's possible that this could be, like we were talking about, that important distinction between if people were having asymptomatic infections in the vaccine group. We just don't know because we don't have that data yet.
That's very helpful. That was kind of what I was trying to get at, which is how do they get these numbers? And it's from what you're describing, it's they had to have some type of symptom to report and say, I think I got something, and go get tested. They're not being, as part of the study, tested once a week or once every 3 days or something. So now let's get on. So we've talked about a lot of my questions for Pfizer already. Let's move on to Moderna. Now, Moderna, they said the vaccine efficacy against COVID-19 was 94.1%. Vaccine efficacy against severe COVID-19 was 100%. So here, Moderna makes a differentiation. Again, this is still their press release. Here with severe COVID that Pfizer does not make. And I'm assuming that's because Moderna has this information and thinks it would sound great and they would get on TV and everyone would be very excited about their vaccine. So then my assumption is severe COVID-19 is that grade 4 adverse event. No, I'm sorry, that's the vaccine. That's just severe COVID-19, puts you in the hospital or ICU.
Exactly.
And so they would measure that the same way. Someone would get sick and call them and let them know. Is that right?
Yes. It also sounds like you'd be a great writer of press releases.
I know, right?
If I read one more press release, I never thought I'd be reading them, but all right.
Yeah, but there's a certain precedent for this as pediatricians. The rotavirus vaccines, especially in global settings in sub-Saharan Africa, where, diarrhea historically has been the second leading cause of kids under 5 worldwide. Pneumonia, number 1, diarrhea, number 2. And so in the rotavirus trials, and lots of the study sites were done in these low resource settings where kids are much more likely to die. Very few kids in this country die of rota, although a lot used to come to the ER. So they looked at like mild, moderate, severe disease, and the rotavirus vaccine was essentially, almost completely protective against severe disease. And 70% versus moderate disease and only 50-something percent against mild. So, which is fine. If kids have a little bit of diarrhea and vomiting, but they don't need any intervention, no big deal, right? That's a win.
That's interesting. And there has never, this is kind of a side question, but I've been thinking about this. There has never been a situation that I have read about where we have created a vaccine and literally tried to vaccinate the entire world at once. Because you have vaccines we've created where slowly first the US and European countries got it. Like, for instance, polio is a great example. There's still areas we're trying to reach. But this is the first time I've read about it. I wonder if you all know of another time where we have made a vaccine and then tried to vaccinate everybody in the world at once, which sounds like a gigantic task.
I feel like the time that would have been most useful is in the wake of 1918 flu, but we didn't have a flu vaccine yet, so this wasn't something that we were able to do. Maybe pandemic flus later on, but you're right. I think that this is a new challenge.
Well, and it's there's unique opportunity now, right? Like vaccines, I mean, rotavirus got to low resource settings around the world very, very quickly because, I mean, people around the world at WHO, Global Health Organizations, Gavi, all these organizations, lots of people knew this is needed. And in fact, greater need in low resource settings than here in the US. But it does take time, public health infrastructure, money, stuff like that. I just think, for the record, polio vaccine is now everywhere in the world. The issues are not really the vaccine itself. Polio type 1, wild type eradicated from the world. Type 2. Type 2 wild polio is eradicated. Type 1 is virtually gone. And the big problem now is there's 2 or 3 regions where, due to civil unrest, war, violence, this kind of stuff, you can't get the population adequately vaccinated. But you're totally, so we're close to eradicating polio. We'll get there. But you're totally right. This really is, I think, unique to say within a couple of years, it's going to roll out to the whole world. And there are organizations, global organizations, working very hard to get it to low resource settings that otherwise, just in terms of profit, like those countries are going to be much lower on the list.
Now, what Moderna also says, they continue to be generally, their vaccines well tolerated, no serious safety concerns identified to date. So now what does that mean? Does that mean the grade 3 adverse events and grade 4 are serious? And so they haven't had any reported grade 3 and 4 events?
I would definitely interpret it to say there were no grade 4 events, but I don't know if I can make a call on grade 3 or not. You remember, it was just that John felt bad enough to stay home from work. So yeah, they might just choose not to tell us that in their press release.
Yeah, well, and here's where it's hard to know, and it's the difference between, like I said, the formal scoring. I mean, technically, that grade 3 that I felt like that I stayed home for a day, that's technically, by scoring criteria, that is severe. But we would all agree, like, should we not license a vaccine because John Williams is a wuss and stayed home for a day? Like, no, definitely not. So I think, when they say in their press release, we don't have serious safety concerns, they mean things that to the Data Safety Monitoring Board or to a reasonable person would be like, okay, this is a problem. I mean, the data safety monitoring work I've done for vaccines, a lot of vaccines cause fever in kids in the first day or so. Okay, if 10% of kids have a fever of 39 degrees, that's definitely not a serious. That would be not at all a reason to not license. Whereas if one kid out of 1,000 had a fever of 41 degrees and, had febrile seizures, you'd be like, that's pretty concerning. That would be worrisome. So I think what they consider serious safety concerns are what we would mean in plain English, like something to really worry about.
Okay. It does take some interpretation, though, and that's why the raw data is helpful. And we're speculating, which is fine. It's just it would be nice with a little more information. Now, we're pediatricians. Let's talk about children. So Pfizer has included kids 12 to 17 years old in their vaccine study, from what I've learned. Moderna is in the process of doing that, but has not already done that. And no one at this point has done studies in pregnant women, which is the precursor to children. So do you all, is what I said correct first, before I keep going?
As far as I'm aware, that's correct.
So if they're getting it rolling and Pfizer already enrolling with the kids, reasonable expectation for a timetable for when this will come out, are we talking months?
I mean, if we look back and I'm thinking about the Moderna vaccine as an example, I know locally we started injecting, and John will correct me if I'm wrong, I think in June. And we're kind of learning about the phase 3 data now in November, December. So 6 months or so, right? 5 months?
I could look back and see the date because I was actually the first volunteer in the Moderna trial.
Oh, congratulations.
Not because I'm special or anything like that, but because I've enrolled in my life in a number of vaccine trials, and this one I thought was so important. Okay, I think that's dose 2, September 11th. So I think here we go, August 14th.
Okay. So August to November, a little shorter than we had expected. And I don't know how large the teenager trial will be, right? So if it's a little bit smaller, it'll probably go faster.
Yeah, they're going to start enrolling. Here at our site, I know they're going to start enrolling soon for the pediatric Moderna. And the reason I know that is both my wife and I were in this Moderna trial, and our kids are registered to be in the pediatric trial.
Oh, now, and for the kids under 12, so younger children, I haven't seen anything about them if they're going to start enrolling them. Is it that if these vaccines are safe in 12 to 17-year-olds, automatically assume it's okay in younger kids, or does there have to be a trial in them as well?
Usually there would have to be a trial in them as well. Yeah.
Okay.
It's very, very, very rare that you would go outside of an age range, especially going into younger kids, especially knowing that kids typically don't get real severe disease.
Right.
And
I think that's the trick is for the vaccine developers, which makes sense. Kids really do get less severe disease. We don't totally understand. I think their function as spreaders fully, but to get this vaccine out to the older adults, especially, and the healthcare providers who are going to get more serious disease and more serious consequences, was the right thing to do. But this does bring up an interesting question for people such as myself with small children at home. So if kind of a public health question. If I have small children at home, I get vaccinated, my wife gets vaccinated, they cannot get vaccinated. This goes back to our earlier point. Things do have to remain the same in terms of masking and social distancing for at least a certain portion of time because we can function as spreaders and they can get disease. Am I saying that correctly?
This is probably true. Although, as we start to see more people in the community be vaccinated, hopefully we would see fewer cases in the community. And I always like to tell schools that schools reflect the community, right? So we can start to think about how if just there's less community transmission in general, maybe things can be a bit more normal than they are right now when you know that there is quite a high burden of disease in the local area.
Yeah, no, I agree with Megan, but I do think you're right, Tony, that I think when people say, oh, we're going to be doing this for a while, I think that's true, that we're going to be wearing masks for a number of months more. My guess is till next summer, late spring, summer is my guess, because I think it'll take that long to vaccinate enough people. I think there's growing evidence, and Dr. Freeman has done some research in this area. There's growing evidence that not only do young kids not get severe disease, that we know, we really know for sure. Young kids, teenagers, young adults, they very, very rarely have severe disease. I think there's really growing evidence that they also just don't get infected much and don't spread much. And that's really different. I think, and a lot of us in the field really were sure it was going to be the opposite. We mentioned flu before, right? Kids are the incubators and amplifiers of flu. 20% of American kids get flu every year, only 5% of adults. And if you shut down the schools in December, you would abort flu season. You would just cut it mostly off. But that turns out to not really be the case for SARS-CoV-2. So why does that matter? Because at least grade school, the schools should probably be open. Because we have seen that with mitigation measures, there's basically no spread in the schools. And the high schools where kids are spreading it, it's after school in their parties. And this is happening in my kids' high school. Nobody's getting it in the classroom. They're getting it hanging out on the weekends. So as pediatricians, I personally think we should all be advocating for, at the very least, grade schools to be in person with masks and distance. It's safe for the kids. It's safe for the adults. And kids are having really long-term detrimental damage to their education that it would be very difficult to fully recover from.
Now, Megan, along those lines, I want to ask you a follow-up question. We had talked at one of our previous podcast episodes about the ACE2 receptors in children maybe not being fully formed, and that's why initially the theory was they weren't getting as much infection and disease with this virus. Has anything more come out about that? Has that been more elucidated or no?
So one of the cool things about kind of the information during the course of this pandemic is that we learn more all the time. So I can't remember exactly when it was that we had that conversation, but for a while it was just a hypothesis. It was a cool idea I had. Somebody actually looked at it, right? So it's actually been confirmed that children in both their upper airway and also in their lungs have not necessarily a different shape of ACE2, but less of it.
Okay, and that's great. That's at least we know the answer now. All right, let's get some paranoia questions in because patients are asking. I've seen these everywhere, and I want us as physicians to have all the data to be able to say, no, this isn't happening. So the information coming out of Pfizer and Moderna, we talked about them being press releases. We've already discussed, couldn't they be manipulating or straight lying to us about the data? They may be able to lie to the press. They cannot lie to the FDA. They have to give that information. The repercussions, especially in this, I assume, with the whole world watching, would be awful. You guys agree with that?
Fair assessment.
Now, when people are actually saying this, that the companies are going to be sneaking microchips or whatever other things into the vaccine that could harm us in the future. Now, John, I'll start with you on this one. How can we be sure this isn't happening? How can we argue this?
I'm sorry, I had a hard time hearing you because I was putting on my tinfoil hat to block the Martians reading my brainwaves. I guess a couple of things. Look, like people, we just gotta be a little bit more realistic. Number one, microchips aren't that small. I mean, they're just not. Okay. Number two, for anybody who has these sort of conspiracy theories about vaccines or whatever, a couple of things. One, the numbers are there. Vaccines, there's only a few companies that actually make vaccines. There are far fewer vaccine companies than there were 20, 30 years ago because there ain't no money in it. Okay? You spend years developing vaccines, which are really, really hard to develop. And you finally get your vaccine, and then everybody says, oh, by the way, you're giving that at cost to low resource countries, right? And the company's like, oh, dang, there goes all my profit. And so, there's not some huge, powerful profit margin. And in terms of the sort of fear conspiracy thing, if people are willing to be, motivated by rationality, not everyone who believes that kind of stuff is. Look, humans can't keep secrets, okay? So the idea that, like, even if microchips were that small, and you think, okay, the government is in cahoots with the vaccine companies to inject microchips into our arms to track us all, like in 1984, Big Brother. The idea that 3 humans in on such a conspiracy could keep it secret, this is like somebody who's never met a human being, right? If you tell something to more than one person, it's no longer secret. There's just no way that 10, 20, 100,000 people are involved and that it hasn't leaked out. So these ideas are just, we have to call them what they are. They're just nonsensical, period.
Megan, I don't want to leave you out. Anything you want to add to that?
I just say that after watching The Social Dilemma recently, I'm much more concerned about, my iPhone tracking my every movement, and, Instagram knowing what kind of products I like, right? Because if you're not paying for the product, you are the product. This doesn't happen with vaccines. I think that perhaps our worries are a little misplaced.
Well, right. This is actually a great point, is that, people who are up in arms about, government surveillance are doing so on Facebook, Twitter, Instagram, Google, all these sites, which we actually know, and we know because nobody can keep secret because we're humans, right? So it's not secret, but we absolutely know that these companies are 100% tracking us. Alexa is recording what you say. Siri is recording what you say. Like, we know this. So
yeah, I've recently seen that documentary as well, The Social Dilemma. It is terrifying, especially with young children. It's terrifying.
Another really good one, kind of like that, is called The Great Hack.
Okay.
Yeah.
I'll have to see that one. All right. That's really what I wanted to know. Did we miss anything? Should we have covered anything we did not?
to me, the only other thing, Tony, is that, like I said, I mean, I believe in vaccines, and we registered for these trials because we think it's that important. I actually don't think I got vaccine. I'm sure I got placebo because I never felt a thing in my arm. I don't care because, the importance is to be in the trial. But, I know a lot of people are saying, well, okay, 40,000 people and we have, a few months of safety data. You're right. But as of this moment, 280,000 Americans are dead of COVID, and another dies every 2 minutes. So you're right. If we wait another year, we'll have a lot more safety data on the vaccines before we widely use them. And 500,000 Americans will have died if we wait a year. So I just feel like, you kind of can't wait. People, it's a pandemic. This virus that nobody had heard of a year ago is going to be the number 2 or maybe even the number 1 cause of death in 2020 in America. Virus we never heard of. It's already number 3, and it's likely to edge into the number 2 and maybe even the number 1 spot. That is staggering. I mean, if that ain't a public health emergency, what is?
I couldn't agree more. And besides the death directly from it, what we're seeing in the pediatric population across the country is a significant increase in mental health disorders and mental health admissions, in anorexia and bulimia admissions. So it's affecting, what we have to do is affecting everybody all the way around. Megan, anything else?
The only thing I'll add is, people have been asking me my thoughts about safety concerns. And I think the best endorsement that I can give is that I will be taking the vaccine when I am eligible. And I've been telling my family and colleagues to do the same.
I think that's a great ending point. We should all, we will take it. We will recommend people to take it. And hopefully next year we'll be doing a podcast together on something else. It will be great.
On Remember the Pandemic.
Remember the pandemic.
We'll bring our scrapbook. It'll be great.
The worst downloaded episode ever. Remember the pandemic.
Yeah.
All right. Thank you both. I sincerely appreciate your time. Thank you so much for joining.
Always a pleasure, Tony.
Anytime, Tony. It was fun.
Time for our acknowledgments. I want to thank our special guest again. I really do appreciate your time. I want to, as always, thank the UPMC Children's Hospital of Pittsburgh, and specifically the Paul C. Gaffney Division of Pediatric Hospital Medicine for continuing to support the podcast. I want to take the time to remind our listeners that there is CME associated with most of our podcast episodes, courtesy of the University of Pittsburgh. So thank you, University of Pittsburgh. Please just click on the link at the bottom of the podcast, takes you to a web page, create a free username and password, answer some questions that I write that are not hard, and that's it. You get AMA Category 1 CME credit. I want to, as always, thank and acknowledge Dr. Megan Keene Tarchichi, who helps me with everything. But I really want to thank you all who are listening. Thank you for letting me come into your earbuds or your car, wherever it is you are. I really appreciate the fact that you do that. If there's anything you want to hear or think we can cover better, any comments, suggestions, compliments, whatever you want to do, again, as always, please feel free to email me at tony.tarchichi, tarchichi, @chp.edu. Thank you again, everybody. I hope you have a great day. We'll see you next time.